Bismillah ar-Rahman ar-Rahim. The process of translation cannot be started until the required components are present. These include all the amino acids that are found in the finished product and, and the necessary machinery. All the amino acids which are going to be present in the synthesized protein must be available when protein synthesis is taking place. In case if any amino acid is missing, the process of translation will stop. Each amino acid has its own specific transfer RNA. Some amino acids which have more than one codon may have more than one transfer RNA. In humans, there are 50 types of transfer RNA. Transfer RNA molecule has an amino acid attachment site at its 3' end. The carboxyl group of the amino acid forms an ester linkage with the 3' hydroxyl group of the ribose of the nucleotide in the CCA sequence at 3' end. When an amino acid is attached to its specific transfer RNA, the transfer RNA is said to be in charged state, whereas in absence of amino acid it is uncharged. Each transfer RNA molecule contains a 3 nucleotide sequence called anticodon at its rounded tip. This anticodon transiently forms complementary base pairing with codon on messenger RNA as it donates the amino acid it is carrying to the growing polypeptide chain. 20 amino-acyl transfer RNA synthetases are present, which catalyze the attachment of specific amino acids to its corresponding transfer RNA, also called the isoaccepting transfer RNA. Each synthetase recognizes a particular amino acid and all of the transfer RNAs that carry that amino acid. For the formation of amino-acyl transfer RNA, the amino acid is first activated by reacting with ATP to form amino-acyl AMP. The amino acid is then transferred from the amino-acyl AMP to transfer RNA. The process of protein synthesis requires a template. Messenger RNA serves as the template with the genetic code, which specifies the sequence and the type of amino acids which are inserted in the protein that is being synthesized. Transfer RNA present in small ribosomal subunit forms complementary base pairing with the template messenger RNA molecule. It ensures that the correct anticodons form complementary base pairs with the right codons on messenger RNA. The large ribosomal subunit catalyzes the formation of peptide bonds in the growing polypeptide chain of the protein being synthesized. The ribosome contains A, P, and E sites. These sites cover both small and large ribosomal subunits. Together, these cover the three neighboring codons present on the messenger RNA. During translation, the transfer RNA carrying the new amino acid, which is to be added to the growing polypeptide chain, comes and attaches to the messenger RNA codon present on the A site. So this codon in its genetic language tells that which amino acid is going to be added. The transfer RNA to which the growing polypeptide chain which is being synthesized is attached is called peptidyl transfer RNA. It is present on the P site. The E site is the empty site. On it the transfer RNA molecule which has donated its amino acid it was carrying to the polypeptide chain which is being synthesized and now it is about to leave the ribosome is present. The ribosomes may be present in the cytosol or attached to the rough endoplasmic reticulum. The proteins which are destined to be exported or form part of the membranes or enter lysosomes are synthesized by ribosomes attached to the rough endoplasmic reticulum. In contrast, the proteins that are required in the cytosol, in the nucleus, in the mitochondria, or in peroxisomes are synthesized by ribosomes that are present free in the cytosol. Though mitochondria can synthesize their own proteins, but most mitochondrial proteins are coded by the DNA present in nucleus and synthesized in cytosol and then exported to the mitochondria. Protein factors are required for initiation, elongation, and termination reactions in protein synthesis. Some of these catalyze reactions and still others stabilize the ribosome, the messenger RNA, and the transfer RNA. G proteins also serve as protein factors. Translation is a major consumer of energy. Addition of one amino acid to the growing polypeptide chain requires cleavage of four high energy bonds. Out of these, two high energy bonds are cleaved during the amino acyl transfer RNA synthetase reaction. First bond is cleaved when pyrophosphate is released from ATP, and the second bond is cleaved when the pyrophosphate is hydrolyzed to two inorganic phosphates by the pyrophosphatase. Two more high energy bonds are cleaved from GTP during the binding of charge transfer RNA to A site and second during the translocation step. Besides that, additional ATP and GTP molecules are required for initiation in eukaryotes, whereas additional GTP molecules are required for the termination in both prokaryotes and eukaryotes. Correct base pairing between the messenger RNA and the charge transfer RNA is necessary for insertion of amino acids in correct sequence in the protein being translated. The charge transfer RNA base pairs with codon and messenger RNA in a complementary and antiparallel manner. It reads the codon in 5 to 3' direction and forms complementary base pairs with it in 3 to 5' direction. There are 61 codons for the 20 standard amino acids that take part in protein synthesis. Some amino acids have more than one codons. Specific transfer RNA is present for each amino acid. 50 amino acyl transfer RNAs are present in humans. A transfer RNA molecule may recognize more than one codon for a specific amino acid. It can be explained by Wobble hypothesis. According to Wobble hypothesis, when anticodon base pairs with codon, the first two bases of the codon follow the rules of complementary base pairing, according to which cytosine always base pairs with guanine and adenine always base pairs with uracil only. But this rule is not strictly followed for the last base of codon towards the 3' end. Here there may be non-traditional base pairing during which uracil may base pair with not only adenine but also guanine if situation arises as shown here. Likewise, guanine may base pair not only with cytosine but also uracil if need arises. Hypoxanthine, which is produced by deamination of adenine, may base pair with cytosine, or it may base pair with uracil or adenine. This non-traditional base pairing becomes possible because the position and depth of the first base of the anticodon in the three-dimensional space is not as precisely set as the other two bases. Therefore, when the transfer RNA arrives and binds, it staggers or wobbles. This movement at the first base of the anticodon allows non-traditional base pairing with the third base of the codon. As a result of this wobbling, 61 transfer RNA species are not required to read 61 codons that code for the amino acids. In prokaryotes, the process of translation and transcription are temporarily linked. Because the process of translation starts without waiting for the transcription to complete, as there is no nuclear membrane in prokaryotes. Whereas in eukaryotes, the messenger RNA is edited after transcription is complete. After editing, it comes out of the nucleus through nuclear pores, and the process of translation starts once the mature messenger RNA has entered the cytoplasm. The genetic code is contained in genes which are present on non-template strand and are transcribed into codons on the messenger RNA, which translates the code into amino acids. The genetic language of codons on messenger RNA is translated into the amino acids, and the amino acids join to form polypeptide, which is synthesized from its amino terminal towards its carboxy terminal. Prokaryotic messenger RNAs have several coding regions. Each coding region has its initiation and termination codon. These are therefore called polycistronic. In contrast, eukaryotic messenger RNA has only one coding region and is therefore monocistronic. Once all the components required for translation have gathered, including prokaryotic initiation factor, abbreviated as IF1, IF2, and IF3, and eukaryotic initiation factors, abbreviated as EIF, the next task is to search the nucleotide sequence AUG or the start codon on messenger RNA. The ribosomal RNA helps in locating it. It is recognized by different mechanisms in prokaryotes and eukaryotes. Example of E. coli can be used to understand initiation of translation in prokaryotes. Near the 5' end of the messenger RNA in E. coli, a sequence rich in purines is located 6 to 10 bases upstream of the initiating codon AUG. This sequence is called the Shine-Dalgarno sequence. The 16S ribosomal RNA has a sequence complementary to all or part of the Shine-Dalgarno sequence towards its 3' end. Therefore, 3' end of ribosomal RNA can form complementary base pairs with the Shine-Dalgarno sequence near the 5' end of the messenger RNA. This helps to place the 30S ribosomal subunit on messenger RNA close to the AUG start codon. Eukaryotic messenger RNA does not have Shine-Dalgarno sequence. In eukaryotes, the small ribosomal subunit containing 40S ribosomal RNA with help of eukaryotic initiation factors EIF4 binds close to the cap structure of the messenger RNA. After binding of the 40S ribosomal RNA near the cap structure, it moves in the 5' to 3' direction along messenger RNA. It continues to move until it reaches the AUG initiator codon. This scanning process requires ATP. Or alternatively, there may be cap independent initiation. In case of cap independent initiation in eukaryotes, the 40S ribosomal subunit, instead of binding with cap region of messenger RNA, binds directly to a messenger RNA region near the start codon AUG at an internal ribosome entry site. Eukaryotic initiation factors EIF4 binds to eukaryotic messenger RNA, and still other eukaryotic initiation factors bind the poly A tail of the messenger RNA. Interactions between the proteins lead to the circularization of messenger RNA. This ensures that incompletely edited messenger RNA is not used in translation because cap and tail are added during editing. When messenger RNA and ribosomal RNA dock together, the next step is the synthesis of the polypeptide chain. For this, the transfer RNA brings the amino acids one by one. To start the synthesis of polypeptide chain, the transfer RNA must identify and locate the start codon AUG on messenger RNA. For this purpose, a special initiator transfer RNA comes into action and finds the start codon AUG with the help of IF2 GTP initiation factors in prokaryotes and EIF2 GTP along with additional initiation factors in eukaryotes. This initiator transfer RNA has unique base modifications which distinguish it from other transfer RNA molecules. In mitochondria and prokaryotes, this initiator transfer RNA carries a methionine at its 3' end. After attachment to transfer RNA, a formyl group is added to the methionine, catalyzed by the enzyme transformylase in presence of N10-formyl tetrahydrofolate, which serves as the donor of the formyl group. However, in eukaryotes, the initiator transfer RNA carries a methionine which is not formylated. In both eukaryotes and prokaryotes, this N-terminal methionine is removed before the end of translation. So here the formylated methionine can be seen. The red triangle represents the formyl group. After binding of the initiator transfer RNA at the start codon, the large ribosomal subunit also joins in. This leads to the assembly of a functional ribosome with charged initiator transfer RNA at P site, which is the only transfer RNA that goes directly to the P site. A site is empty at this time, otherwise all the transfer RNAs carrying the amino acids go to the A site. Certain special EIF prevent the arrival and joining of the large ribosomal subunit before joining of the charged initiator transfer RNA carrying methionine at the start codon in eukaryotes. GTP on EIF2 is hydrolyzed to GDP in eukaryotes. Guanine nucleotide exchange factor EIF2B replaces GDP by GTP, leading to the reactivation of the EIF2 GDP. Now the charge transfer RNA carrying the amino acid for the codon downstream the initiator codon occupying the A ribosomal site appears on scene. This process is called decoding because the genetic code language is deciphered or translated into amino acids. Elongation factors and GTP are required for this process of decoding. The carboxyl group of the amino acid attached to the initiator transfer RNA present at the P site and the amino group of the amino acid attached to the charge transfer RNA present on the A site react with each other to form a peptide bond between themselves in a reaction catalyzed by ribozyme. Peptidyl transferase belonging to the large ribosomal subunit of the ribosomal RNA catalyzes this reaction. After the formation of the peptide bond, the peptide on the transfer RNA at P site or the initiator transfer RNA is transferred to amino acid on transfer RNA at A site. This process is called transpeptidation. The ribosome then moves by one codon or three nucleotides towards the 3' end, whereas the transfer RNAs don't move, they stay where they are. This movement requires elongation factors and GTP hydrolysis. Since the transfer RNAs don't move with translocation of ribosome, after this step the uncharged transfer RNA automatically occupies the E site and the transfer RNA carries the growing peptide chain now sits on the P site and the new transfer RNA arrives to occupy A site. This forms peptide bond with growing peptide on P site. undergoes transpeptidation followed by translocation. The whole process is repeated again and again until a stop codon is reached. As the messenger RNA is very long, more than one ribosome at a time may be involved in translation. This complex of one messenger RNA and many ribosomes is called a polysome. As the ribosome is moving along the messenger RNA, different codons continue to occupy A site until a stop codon occupies the A site. As this occurs, translation stops. As the release factor protein binds to the stop codon, the bond between the newly synthesized peptide chain and the transfer RNA is hydrolyzed, so the newly synthesized protein is released. A third release factor, RF3, then causes release of RF1 or RF2 in presence of GTP, which is hydrolyzed. Though gene expression is regulated at the level of transcription, but translation may also be regulated. One important mechanism for regulation of translation in eukaryotes is through the modification of initiation factors. For example, EIF2 can be modified by phosphorylation. Phosphorylated EIF2 is inactive. So, when EIF2 is phosphorylated, the process of translation cannot be initiated. Not only that, certain proteins may bind transfer RNA and prevent it from translating into protein. After their synthesis, the proteins are folded so that they can become functional. Folding may occur spontaneously or it may be assisted by chaperone proteins which assist in folding, either by binding to the polypeptide as it is being synthesized, or these can act as template to assist in folding. I have discussed these processes in detail on my lecture in my lecture on protein folding. So to summarize, first of all, the initiation factors help in the formation of initiation complex, which contains the small ribosomal subunit, the template messenger RNA, the transfer RNAs and the required amino acids. Then the initiator charge transfer RNA carrying methionine arrives on the P site with the help of ribosomal RNA and initiation factors and forms base pairs with the initiator codon through its anticodon. After this binding, the large ribosomal subunit joins in. In the next elongation, in the next step, the elongation factors bring the charge transfer RNA carrying the amino acid according to the codon occupying the A site, and it joins through complementary base pairing with the codon on the messenger RNA. This process is called decoding because genetic nucleotide code is decoded to amino acid. GTP is hydrolyzed to provide energy for this step. Next, the peptidyl transferase activity of the large ribosomal subunit leads to formation of peptide bond between the amino acids on initiator transfer RNA at P site and the charge transfer RNA on A site. Not only that, it causes the transfer of one amino acid on the initiator transfer RNA to the transfer RNA on A site through transpeptidation. Next, the ribosome slides past the messenger RNA by 3 nucleotides or 1 codon, whereas the transfer RNAs don't move. Thus, now the transfer RNA with growing peptide chain occupies P site. A site becomes empty so that new charged transfer RNA may come and occupy it, whereas the initiator transfer RNA now occupies the E site from where it leaves. GTP provides energy for translocation step. These steps of elongation, transpeptidation and translocation are repeated again and again until stop codon is reached.